this lab, we're shifting from looking at what plant cells are made of to how plant cells reproduce, grow, and pass on genetic information. We'll focus on the cell cycle, mitosis, and meiosis, and you'll be observing real dividing cells under the microscope, as well as using models and images to understand what's happening inside the nucleus. Before we begin the activities in this lab, let's take a look at our learning goals. By the end of this lab, you should be able to describe the major stages of the cell cycle and recognize what is happening during each phase. You will learn to identify and correctly sequence the stages of mitosis and understand how chromosomes behave during each stage. You'll also compare mitosis and meiosis and explain how each process contributes to growth, tissue repair, and reproduction in plants. Finally, you'll practice interpreting real microscope images of dividing cells using visual evidence to support your identification of each stage. As you work through the activities, keep these objectives in mind. They will guide what you should focus on observing, drawing, and explaining. All living cells go through a repeating life cycle called the cell cycle. It begins when a new cell is formed and ends when that cell divides to make new cells. During the cycle, the cell grows, copies its DNA, and then divides. This process is continuous in tissues that are growing or repairing themselves. Interphase consists of the G1 phase, where the cell is growing and monitoring its environment before committing to division. During the S phase, the cell replicates its entire DNA content. The G2 phase is a final period of protein synthesis and growth before mitosis occurs. Mature plant cells exit the cycle and enter a state called G0, where they remain in interphase. In a typical cell cycle, interphase is the longest stage, accounting for 90 to 95% of the cell's total life. Cell division actually happens in two main steps. Karyokinesis is the division of the nucleus, where the chromosomes are separated. Cytokinesis is the division of the cytoplasm, which physically splits the cell into two separate daughter cells. In plant cells, this involves forming a cell plate that becomes the new cell wall. There are two major types of nuclear division. Mitosis produces two genetically identical daughter cells and is used for growth, tissue repair, and asexual reproduction. Meiosis produces four genetically different cells with half the chromosome number and is essential for sexual reproduction. In plants, mitosis allows roots to grow, stems to elongate, and damaged tissues to be replaced. Many plants reproduce asexually, such as the strawberry plant producing runners. If you cut off the runner, it is an identical plant to the mother plant. Each new cell receives the same genetic information, ensuring that all cells in the plant function properly and consistently. Meiosis is necessary for sexual reproduction. It produces cells with half the normal chromosome number so that when fertilization occurs, the resulting plant has the correct chromosome count. Meiosis also introduces genetic variation, which is important for evolution and adaptation. Genetic variation may produce new cultivars of hibiscus like shown in this image, or it may produce plants that are resistant to a disease or can withstand warmer summers. Before mitosis or meiosis begins, the cell passes through interphase. This is when the DNA is copied and the cell prepares all the materials that it will need to divide. Even though interphase is not part of mitosis, it is essential because without DNA replication, DNA could not occur. In this lab, you'll be observing cells that are actively dividing, which means you'll see different stages of the cell cycle frozen in time under the microscope. You'll identify cells in interphase, where DNA is being copied, and in the stages of mitosis, where chromosomes condense, line up, separate, and are pulled into two new nuclei. In some activities, you'll also examine the stages of meiosis and compare how chromosome behavior differs from mitosis. As you work through the lab, your goal is to use visual evidence to recognize each stage, describe what is happening to the chromosomes, and connect these observations to the roles of cell division in growth, repair, and reproduction.